Hi.
Hi, Bradley.
Hi.
We woke up late today.
Oh, yeah.
No, I had just you last week on the breast cancer stuff. This week I had two people. So
okay.
It's an exponential growth.
Yeah.
Okay. Yeah, I woke up, I was dreaming all night about addressable X-ray curve. Up in the chat.
Great. Hey, Susan.
Hi, I made it.
Yeah.
Okay.
Addressable X-ray sources. Okay.
Very good.
I sent you an email, and one of the pieces was too long. I should have just sent you the link. So my apologies for that.
So,
yeah. I copied cell mobility YouTube video. It's sort of a group that get together and present their work.
All right.
So it's interesting, and it's just a YouTube channel that I found interesting.
Oh, okay.
And I sort of took notes by copying the one person's slides because it was on mechanics.
Okay. Good. Okay. When did you send it? This morning?
I sent it to Bradley.
Oh, yeah, I did get it. Yeah.
Okay.
So there's a YouTube video. Maybe I'll just find it. It was good. Just need to find it. It's here somewhere. Okay. No, not that one. Oh no. Okay, that's going to take me a minute. It's all right. I'll just post it when I find it.
Okay. Yeah, that's good. So yeah, it's great. I don't know if anyone else is going to show up for the meeting, but I'm not sure. Yeah, Susan, you sent along some papers in that batch too. That looked pretty interesting.
Yeah, I have a whole folder that I've called, what do they call it? Amoeboid to ramiform.
Yeah. All right. Well, yeah. See if you can find that paper. Oh, there it is. Okay.
Wow.
That's just the YouTube. That's the YouTube channel on cell migration.
Oh, right, the cell migration videos. Yeah, I've seen those.
Yeah. And what got me into this is the blood types that change. You add, what was it? A cytokine to the mix, and then you centrifuge it, and you get amoeboid and ramiform white blood cell types.
Oh, wow.
Anything that centrifuges something, I just always think it's really great that they're alive still.
Yeah. That they're intact, they don't come apart.
Yeah.
Yeah, there are different techniques for that. You have to be careful. Sometimes you have to if you do a really low centrifuge speed, you can recover a lot of things.
Yeah, I had a hand centrifuge, and then Dick gave
also 10 minutes is a good time to keep in terms of, like mine is also, I think, around 20 or something.
Anyways. I bought a little centrifuge that I still have, and I have a hand one, so you can on YouTube. All right, so we'll just always 10 minutes is a good time. Yeah, we'll do our usual, like mine is also 10 minutes. Okay.
And get
Have you got the YouTube on?
After the hour. So, Ashwin, how's Stanford? Are you enjoying California after all those years in Wisconsin?
Well, it's fun. I'm
Yeah, so
Okay, well,
whose audio is it?
audio is getting through, yeah.
Oh, okay. Oh, I think that was
Okay.
maybe Dick.
All right.
But yeah. So I guess let's see. I'll get started. I had a couple of features to show. Let me see. I'll share my screen. I don't know if anyone else is going to show up, but we'll start.
Okay.
All right. So this is.
I always find this is a great way to start my week.
Oh, good. Yeah.
Yeah. So I'll shut off my audio. So.
Oh, okay. So this is the first thing I'll talk about is this. This actually comes from a conference on developmental biology, and this is sort of reproducing natural structure in organoids and in embryos. So this, I don't know if I can zoom in on this, if I hide that probably. So this is a picture of the hourglass model of development. So this is where if you start development at the bottom, you have your eggs, and they start to differentiate into the different parts of the mammalian embryo. And then as they move forward in time, you start to get what they call Hox gene expression, and you start to get these segments of the embryo. But there's this period called the phylotypic period, which is this hourglass. So the number of pathways that the embryos can take as they as the animal species are very, somewhat large in this space down here. But then in this phylotypic stage, every embryo from fishes to humans to mice, they get restricted in terms of how they express their Hox genes in the sort of the form of the embryo. So these embryos look different at the bottom. They have very diverse pathways to getting to this stage, which is the phylotypic stage, where they all kind of look similar, and they have this sort of segmentation that starts to occur where you get the head and the tail and the midsection. This process is very similar. And then as you move out from the phylotypic stage, you move towards different phenotypes, like a rat and a rabbit and a mouse and a human and so forth, and a fish. And so there's a wide variety of phenotypes on this end of the hourglass. But this part, they all look somewhat similar in their phenotype.
Okay.
What's the reference for that?
This was in Dubole 94. So this is just an example. I mean, this is an hourglass model that people have proposed. This is where they cite this from.
Yeah, let me give a challenge to that.
Okay.
Are you familiar with mosaic embryos?
Yeah, mosaic embryos.
Which are made from different species.
Okay, yeah.
Okay, so how would that fit into this scheme?
Well, if they're two mammalian species that you've created together, they basically go through the same phylotypic stage. I don't know what would happen back up here where they start to make their own.
Suppose you do a mammal and a mammal and a fish.
Okay. Vertebrate fusion or fusion of different vertebrate types. I guess they go through the same process. I don't know.
I'm just wondering to what extent this is hand waving and not a real explanation.
Well, I think this is like a theoretical model that people have observed from.
Well, it's
Yeah, I know it's.
It's okay, fine, but what's the mechanism?
Yeah. So that's the hourglass model, and it's
okay, but maybe maybe the thing to do is go back to the old literature on mosaic embryos and ask what on earth's going on in terms of this model.
Yeah, I have to look that up. Yeah.
Okay, because those papers are probably from the 30s.
Yeah.
I think part of this is they all have a beating heart when they go through the pinch in the hourglass.
Yeah.
No. You have to look up Steve Smith's work. on whether or not you need a heart, because he had a heartless axolotl.
And it still went through the hourglass?
It still produced an axolotl, yep. and it seemed to breathe without a heart.
Okay.
Yeah, he used to give a lecture called "You got to have a heart. You got to have heart, or do you?"
Yeah.
Yeah.
Yeah. So this, yeah. So this chart actually is interesting too. This shows pre-gastrulation and early gastrulation, and this shows, I think, mouse embryo, and this is comparing like what they call a gastroloid versus an organoid. So this is an organoid when they have the flat sheet here, and the gastroloid is this thing that has shape to it. And so they're showing that you could get these different segments of different cell types, and you get movements in different directions. So we were talking last week about how you can form like a tube that curls around, and this just shows like the direction of cell movement that's kind of recapitulated in some of the embryoids versus in like something that approximates an actual embryo. So this is where you have pre and early gastrulation, and those patterns continue, and they kind of move in a certain direction. You see that in chick as well, where there's movement outward in a flat sheet, but in this thing that has shape, it moves in the same direction, but it's a little bit different in terms of its shape. And so you have these different things that you see in different embryos. It's a kind of an interesting figure. I don't know what to make of it beyond this, but I just thought this was an interesting figure. And then this, I guess, yeah. Oh.
Bradley, was there any way of getting some crude differentiation tree for those different gastrulations and see if they're similar or not?
Yeah, I guess we could.
Okay, because they're showing these nicely divided, separated tissues without discussing why they're different.
Yeah.
Okay. As to the beating heart thing, I maintain that when you have a heart that's pushing things around, it changes the mechanics of the tissue because it makes it stiffer. So that's going on with some of these things.
Yeah. Well, it's possible. Yeah, I mean, I don't know. I mean, that's a different question about like the mechanics of like what the heart brings versus what just kind of the mechanics are maybe of different tissue types. I mean, I don't know. I'm not an expert at it, but all right. So I mean, I thought that figure was kind of interesting, and so the next one I want to talk about is this. This is a nice article. This is experimental Lamarckism, and this is out of American Scientist. This is an experiment. This is Brian Hayes. I don't know. He's a writer for American Scientist, and he did this really interesting experiment where he asked, would this evolutionary strategy be worth the investment? So Lamarckism, of course, is this idea from Lamarck where if you have, say, like an animal that wants to sort of increase its neck length, like a giraffe, or wants to increase its muscle mass or something, it just behaves, and that behavior reinforces increases in the size of the structure. And so that's how Lamarck proposed that the giraffe had acquired such a long neck, because the giraffe went to eat leaves off the trees, and it kept stretching its neck upward and upward, and then it finally ended up with a long neck. And of course, Darwin knocked that idea down because, first of all, there was no hereditary reason for it, or no hereditary mechanism for it. But most importantly, it didn't seem to square away with a lot of the evidence that you saw that better fit was better explained by natural selection. But people have used this idea of Lamarckism in computational modeling, in genetic algorithms. They've used Lamarckism a lot to sort of build algorithms that do certain things. So the reason I mention that is because this is a computational experiment comparing Lamarckism with Darwinism with actual learning algorithms. And so it's an interesting comparison. So, Lamarckian ideas existed. They were pre-Darwinian, and they were eventually kind of went into disrepute. And, people know what it is, but it's like, what is it really explaining? So, but really one of the things that Lamarck kind of focuses on is this idea of feedback loops. And so the trouble with, all right, so they say they have these questions about Lamarckism, and so one way of answering them is to note that molecular pathways needed for Lamarckian inheritance just don't exist. Within the context of life as we know it, there's no way for the elephant's nose to talk to the elephant's genes, especially the germline genes. The central dogma of molecular biology says information flows from DNA to RNA to protein, not the other way around, which would be required for Lamarckism. A Lamarckian feedback loop would seem to require some mechanism by which the proteins of the phenotype could alter the DNA of the genotype. So, but then that invites a question as to why is it that these feedback loops never evolved? So it's not impossible that those feedback loops could have evolved at some point, but they never did. And there are a lot of shortcuts and genetic detours like plasmids, transposons, retroviruses, prions. It seems a bit arbitrary to declare this one pathway out of bounds. So, other than like there are obvious things about the central dogma and the Weitzman barrier that hold, but why have we not seen the evolution of something like this? And so maybe another possible reason why we don't see Lamarckian inheritance is it's not worth the bother. So, it's just not worth the organism's time to evolve this pathway. And I say that like as sort of an anthropomorphic thing, but what he does in this paper is he introduces this set of simulations and goes over a number of days, learning in Lamarckian and then Darwinian. And he goes for 500 days on learning in Lamarckian because they're faster processes, but goes to 2,500 days in the Darwinian case. And then he has this camouflage phenotype, which actually matches an example you see often in that sort of demonstrates natural selection. And this is the peppered moth example. And so if you're not familiar with the peppered moth example, it's that you have these moths that were in pre-industrial England, and they had different wing colors. And then when industrialization happened, there was a lot of coal burnt and a lot of soot on the branches of the trees, and the darker morphs were able to camouflage better with the soot, and they were able to survive at a greater frequency. So there have been some people have revisited that experiment to show that that's not exactly what was happening, but this is, he's using a similar model here, and for this purpose, I think it's fine. So there's this camouflage phenotype that he uses. It's lighter or darker, and the idea is that if you run this simulation for so many days using these types of transmission and feedback, this is what you get. So you end up in the Darwinian case, you end up with these straight lines, and these are really just frequencies of the phenotype. So the lighter phenotype appears early, the darker phenotype appears early, then the darker phenotype disappears, and I assume that the selection is for lighter phenotypes because this trend line is going upward. So it goes from like a median value up towards lighter. And so the darker, in Darwinian evolution, the darker phenotypes disappear gradually, but not entirely. You do get some that are maybe a little bit above the mean, and then these lighter phenotypes at the very top disappear, but everything tends towards this line, although it's a little bit below the line. But you see that you have these frequency bands that are sort of the result. Now that's important. When you go to learning in Lamarckism, or Lamarckian evolution, you see something really interesting. You have the same trend line that is basically the same trend from median phenotype to the top. You have these, but instead of having these frequency bands, you have this sort of convergence upon this band or this line as you go up. So in learning, it's basically learning that this is the desired phenotype, and everything goes into this direction. So you get lighter and darker phenotypes that converge upon this line. In Lamarckian evolution, you get a similar thing, actually, than what you would call learning. And I guess learning would be like standard, like learning of, maybe like a Hebbian learning or something like that. You get this convergence to this trend line, but you also get these, like, I guess I don't even know if they're frequency bands or just kind of streaks that come out. So there's a little bit more variation in some ways in learning, maybe not, because you get the same thing in learning, but it also converges to this line. And so what that suggests is that Darwinian evolution is actually producing, it's moving towards that fitness, but it's also leaving a number of phenotypes that are sort of not so tightly selected for. And the reason that's important is because you want to be able to, if you're evolving a trait, you don't want to get stuck in that place. You want to have enough genetic diversity to move to another fitness imperative later on. As say, you have soot on the branches of your trees, and then that soot goes away. Then you don't want to have, you don't want to go to extinction because you can no longer blend into the tree branch. So that's the idea here. And so learning in Lamarckism or Lamarckian evolution will sort of converge upon an optimal solution, but that optimal solution comes at the expense of this ability to adapt to later changes in the environment. So that's the way I would interpret this. And so maybe that's why we have, maybe that's why nature never bothered to evolve a Lamarckian mechanism, or better yet, why natural selection is the thing that we ended up with.
Hey, Bradley?
Yeah.
I put a book by Arthur Kessler in the chat,
Okay.
Which is exactly on a supposed case of Lamarckian evolution.
Okay. Yeah.
Okay.
All right.
I don't have the book.
All right.
Okay.
Yeah.
But Kessler was an interesting character. I think he might have been right about some things, and this is his examination of a case of Lamarckianism.
Okay. Yeah.
Okay.
Yeah. Thanks.
Okay, I think this is the case that drove the discoverer mad. Because he wasn't accepted.
Oh, right. Yeah.
But you have to remember, neither was plate tectonics.
Right. Yeah.
Okay.
Yeah.
So,
Actually, in this case, he actually does model with, so this example from the peppered moth is called melanism, and it's, yeah. And so they actually use a star logo program, which is an agent-based model for this. So this is the main actors in the star logo program are mobile animal-like objects. Then they use these as these moths, and they show the population here where actually it's interesting that the population here over a certain number of days, it goes from 400 and it goes down. What is this example though? It's showing that there's an increase in the Lamarckian case in terms of the population size, but a decrease in the Darwinian case, and it's sort of a wash for the learners. It's a little bit less, and so this is actually, this experiment is where he's modeling melanism as a polygenic trait. And so the moths have 3 other genes which range in value from 1 to 1,000. We gave the genes names kudzu, Harvard, and Vanderbilt. Kudzu is a growth gene, and kudzu is linked to the Harvard gene, which governs the rate of learning, and the Vanderbilt gene, which controls an inheritance of acquisitions. So this is more about sort of these modeling sort of a circuit for for Lamarckism to work. And so this gives you this sort of feedback. So let's see. In the extreme case of perfect Lamarckism, the color gene of the offspring is set equal to the parent's color phenotype. Lesser degrees of Lamarckism interpolate between the original genotype and the acquired phenotype. And so this is a test of Lamarckism, and this kind of shows this melanism is Lamarckism and how the Darwinian population crashes, the learners are kind of at a moderate level, and the Lamarckian population goes up. And so it just shows you that those dynamics of that simulation. And so it's an interesting little article. He does a lot of really interesting experiments in this. Yeah. So, we have any questions about that, or see Jesse's here? All right. I don't know if you wanted to say hi, or you can say hi in the chat, I guess. So the next thing I want to talk about is we talked about this several weeks ago, and this is a paper that I have kind of prepared for this special issue of the it's this Symmetries, Minds, and Matter, I believe is the name of the special issue. And I got an email from the person who's organizing this special issue. And they wanted me to submit something from our group. So I've been working on this manuscript, and it's almost ready to submit as a first draft. It's not really, I just wanted to get some feedback on it. So basically this article is about looking at development and symmetries in the evolution of nervous systems, or the development of nervous systems, and how some of these things can be modeled. So in this paper, I kind of go through some of this. I have 3 examples, one being tensegrity, one being using differentiation trees, and the third using these embodied hypergraphs that I've been talking about this past summer. So this is, these are 3 ways to look at this type of system. The system's defined. It's a developmental system that generates structure, and then it generates these nervous systems, these nervous system networks, and it's all defined in terms of cognition and some of the things we can learn from embodied cognition specifically. And so the paper kind of goes through building that model, and then bringing in these 3 models. There are these 3 types of analysis to look at like the different aspects of this, and then finally, end up with some other like sort of insights, what we can learn from it. So this is due by the 31st. I'll send out a copy of it for people to look at if you want to, suggest changes or things like that. It's pretty much close to the word limit. It was like 8,000 words, I think, is the limit. The target journal is the Royal Society Interface Focus, which is a interdisciplinary journal. And they're going to have a number of different papers on different topics related to symmetries and mind and matter. And I think they're going to have a couple of things on biological symmetries, which are what's talked about in this paper. But yeah, I think it's a little bit different than the rest of the papers, but that's probably okay. I think that's what they wanted to pick our group for. So
I'll look forward to reading it.
Yeah, I'll send out a link with the document in it, or I'll attach the document to it. So yeah. Okay. Let me bring my thing back up. Any questions or comments at this point? Okay. All right. So let me see. Where are we? Oh. Right, so actually, Susan sent me some papers on cell contractility and polarization, and for some reason it's going back here. And so I was going to move on to that. So yeah, Susan's been sending some nice papers on cellular physics and cell contractility, and so I figured I'd cover some of these papers, and we can talk about them. So this is one of the papers. I think this has to do with what she was talking about with having a heart, well, at least a little bit maybe. The dynamics of microglial polarization reveal the resident neuroinflammatory responses after subarachnoid hemorrhage. So this is a paper on neuroinflammation plays a critical role in the pathogenesis of subarachnoid hemorrhage. Is this a paper you recognize, Susan? You sent me an abstract on this. I don't know if she's still available, but anyways, microglia is the resident immune cells orchestrate neuroinflammation distinctly in neurological diseases with different polarization statuses. However, microglial polarizations and the neuroinflammatory responses after SAH is not fully understood. So they use this. They use this model here. We found that reactive immune cells were largely resident microglia pool rather than infiltrating macrophages. So this kind of goes through this model. Microglia polarized dynamically from M1 to M2 phenotype, along with the morphological transformation from ramified to amoeboid shapes. So this is, I think, what she was talking about with the amoeboid stuff here, where she was talking about, yeah, so this is where you have, okay, so microglia polarized dynamically from M1 to M2 phenotype. So they have these two different phenotypes, and I think they're related to just their basic shape, along with the morphological transformation from ramified to amoeboid shapes. So they're transforming their shape.
Yeah, so this was with microglia, and then there was the ones with blood.
Okay. So what is the takeaway from this paper? Or do you know, or did you read it?
No, I didn't read this one. I send you the reference? I was having trouble getting it.
Yeah, I was able to get it. Yeah.
The problem was. But yeah, it just, what I was interested in is the fact that it transforms from ramified to amoeboid shapes. And when you're doing brain development, you get amoeboid shapes that turn into ramified shapes. Like, so this is going on, with the brain.
Yeah.
What does ramified mean?
Long and thin.
Oh.
Branching? Yeah.
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 That's right. The flat pancake to long, thin branching thing.
Yeah.
Yeah, I don't know.
Yeah. Yeah. So it was interesting here is that bipolar shaped microglia appeared as the intermediate and transitional status with the capacity of bidirectional polarization. So they actually have this very interesting morphogenesis, I guess you could say. Did they mention in this paper why that is? Is there a functional reason for it, or?
Oh, like I said, I think that was just a reference that I sent you because I was having a fight getting the paper for some reason.
Okay, well, I'll send you the paper, yeah.
Okay.
Yeah. The microglial polarization status is distinct in molecular inflammatory responses. M1-related pro-inflammation was predominant in the early phase and subsequently transited to the M2-related anti-inflammation. The systemic characterization of dynamics of microglia polarization contributes to the understanding of the origin of neuroinflammatory responses. And so this is basically a response in terms of like inflammation or some chemical signaling is influencing the shape of these glia. And then that's influencing sort of the nature of how these things are regulating some of this hemorrhage, when this hemorrhage occurs. So see if they have any examples of, they show kind of this region of the brain in this picture, and they show the hemorrhage site. I don't know if they show the cells. That's what I'm looking for. So they just show kind of a slice of cortex. It's not that interesting. They're identifying it through immunofluorescence. Okay, this is an example of the cells, ramified versus amoeboid. So these are the amoeboid cells, these are the ramified cells. So they have these branches coming off versus amoeboid, which is this flat pancake-like thing. So microglia accumulated morphological transformations after SAH. So a ramified glia, microglia, kind of goes out and branches out towards the neurons and is a support cell. And then this amoeboid shape is actually something that looks like it's maybe not as active, or not doing its job after it takes on this shape. So it's like this flat pancake, like a fried egg almost. And so it's losing its branches that go off like this. So that's interesting. And this is an example here of the shape from day 1 to day 10. So in day 1, you have these branches that go out towards neurons and other parts of that part of the brain. And then you get this, almost looks like these irregular branching structures. And then you have this single branch structure here. It's not really even branched anymore. It's just kind of like a big mass. And then you get these almost look like blebs or amoeboids in there, where they're just not necessarily doing anything. They're just sitting there. Yeah, so that's an interesting paper. I'm not too familiar with that system, so I can't say any more about it. It's just interesting how there's that change in cell shape as you get that sort of a problem in the brain you get. So this paper then is coordination of contractile tension and cell area changes in an epithelial cell monolayer.
Okay, this is was on that YouTube video.
Okay, yeah.
This is the author.
Yeah, this is the...
explained it on the YouTube video.
Okay. So that was in the... Yeah. So let's go through the abstract as usual. During tissue development and repair, cells contract and expand in coordination with their neighbors, giving rise to tissue deformations that occur on length scales far larger than that of a single cell. The biophysical mechanisms by which this contractile force, or these contractile forces of each cell, cause deformations on multicellular length scales is not fully clear. So to investigate this, they start with the principle of force equilibrium, which dictates a balance of tensile forces between neighboring cells. Based on this principle, we hypothesize that coordinated changes in cell area result from tension transmitted across the cell layer. So this is the cell area. Well, we saw an example in the last paper of cell shape. Now this is changes in cell area that are coordinated. And this is resulting from tension transmitted across the cell layer. So I imagine there's a tension signal or some signal that controls tension. To test this hypothesis, spatial correlations of both contractile tension and the divergence of cell velocities were measured as readouts of coordinated contractility and collective area changes, respectively. So this is coordinated contractility and collective area changes. That's what they're measuring. Experiments were designed to alter the spatial correlation of contractile tension using 3 different methods, including disrupting cell-cell adhesions, modulating the alignment of actomyosin stress fibers between neighboring cells, and changing the size of the cell monolayer. So these are 3 different ways that they did this. Cell-cell adhesions are, of course, the things that keep cells sort of in contact or together, and that can introduce, of course, stresses and tensions across different cells as they're pushing against one another and sticking to one another. Modulating the alignment of actomyosin stress fibers between neighboring cells, so the stresses are transmitted, I guess, across the different cells in a coordinated manner. And then changing the size of the cell monolayer. So this is where the size is bigger or smaller. In all experiments of spatial correlation, both tension and divergence increased or decreased together. So this is tension and divergence in agreement with our hypothesis. I guess divergence is either that the cells are coming apart, they're adhering to each other less. To relate our findings to the intracellular mechanism connecting changes in cell area to contractile tension, we disrupted activation of extracellular signal-regulated kinase, or ERK, which is known to mediate the intercellular relationship between cell area and contraction. Consistent with prior knowledge, a temporal cross-correlation between cell area and tension, which is over time, of course, revealed that ERK was responsible for a proportional relationship between cell area and contraction. So as the cell area increases or decreases, contraction increases or decreases. Inhibition of ERK activation reduced the spatial correlations of the divergence of cell velocity but not of tension. So this is where ERK is inhibited. The spatial correlations between divergence of the cell velocity is maintained, but not tension. So together our findings suggest that coordination of cell contraction and expansion requires transfer of cell tension over space and ERK-mediated coordination between cell area and contraction in time. So these cell contraction needs to be coordinated, and the expansion requires transfer of cell tension in space. So when you have cells in a cell population in a culture dish or in a tissue, those sorts of things need to be, cell contraction needs to be coordinated, and the contraction and expansion of cells requires a transfer of cell tension over space. So tension needs to be coordinated across the cells. And then ERK-mediated coordination, which involves this molecule ERK, there's a coordination between cell area and contraction in time. So this controls the temporal aspect of it. So let's see if we can find some, so these cell culture for this, these micro-patterning to introduce or to use it as a substrate. So micro-patterning is where they put a pattern on the culture dish, and they allow it to, they have like conditions where it's adhering to the surface, so they can control those conditions. Then the divergence of cell velocity, they have some equations for that. So they're actually looking at divergence as cell velocity, and they use these measures. They have a spatial correlation, of course, temporal cross-correlation, and all that. So let me see if I can find some images. So this figure is where they disrupt cell-cell adhesions and observe decreased spatial correlations of contractile tension in this div vector. So this is where you have the cell, or the, let's see, this color map is kind of catching my eye here. So this is where you have, okay, so the first one is root mean squared traction and control cell islands, and islands treated with this thing that they're treating it with. So you have this is in pascals. So you can see that traction is increased in the control here, and more increased in the control. So average tension versus root mean squared traction. Tension is higher. Then you have tension correlation between the control and the treatment. The correlation is less than the control here. You can see that the correlation length is higher for the control than the treated. Actually, this correlation actually extends over a shorter distance scale, is what's happening here. So it's less than the control, but it's also, and then it kind of catches up at longer length scales. So I don't know if there's any interesting stuff with the length scale going on, but then this is of course E and F, or where you have these different islands that are treated. So you have the control versus the treated, and you can see it's a mosaic. It's not exactly in one spot versus another. So they're in different patches in the culture. And then G and H are these spatial correlation of the div vector of control in EGTA treated islands. So this is divergence correlation here, where they show that the divergence correlation for the treated lags behind the control, but then increases at about 250 microns. And then you can see this correlation length scale difference here in this graph. When they do a graph like this, they usually give the observations as these dots, and the line is like the median or the mean. So just so you know what's going on there. In this case here, these two figures here, B and C, are confocal images of stress fibers of low serum and CNO3 treated conditions. So you can see this is the low serum condition, this is the treated condition. And in this case, the traction is much higher for the treated condition. The average tension is also higher. You get this correlation of the treated condition is higher for tension at low length scales, but then falls behind the control at higher length scales. You get a correlation length that's higher in CNO3, and then in divergence correlation is kind of higher in the treated up to 150 microns, and then it falls below the control. And then you get this difference between the treatment and control of low serum and for correlation length. So it's a little bit different. It's actually a different result than this. They're using two different things here. They're using EGTA and they're using CNO3. So this is a Rho activator. So there's a lot of molecular biology here. The difference, I guess, is just the way that which the cells are sort of, the tension is transmitted in the cells. And so I'm just going to go through more. I don't know if there are any other good figures in here, but yeah, I think that's it. So that's an interesting paper. Did we have anything to say about that paper? I don't know. Maybe there was something interesting.
I'm just glad you have shown that it's actually getting into some of the tension in cells, like digging deeper into it, which was nice. And this author has a couple more papers along this line, and he did present all of it in that YouTube video, this most recent research.
Yeah.
So the YouTube video there, the first half of the YouTube video is the author. I don't know if I can pronounce his name. Starts with a capital A, small a, and carries on. Yeah. So I won't try to pronounce his name. They do an excellent job on the YouTube video.
Yeah.
Okay, Susan, I put the papers by Steve Smith and on axolotl heartless axolotls
Yeah.
in the chat.
Okay. Yeah, I got it. Actually, I copied and pasted it.
Okay, good.
So I'll look it up.
Yeah, Steve was a paraplegic and he didn't live long.
Oh, okay.
Yeah, he had a car accident which caused paraplegic. Caused him to be paralyzed.
Yeah, well, it depends on which vertebrae gets hit. Depends. Yeah. Well, I met a quadriplegic.
Yeah, he was.
And I looked after a quadriplegic too, so.
I think he was quadriplegic. Anyway.
Okay. Quadriplegics have a bad time.
Yeah, he didn't. Yeah, he died a long time ago. But anyway, he did get these papers done. As a quadriplegic.
Well, yeah, I'll look at it.
Okay, so today we've talked about groups of cells where you have physical changes that change their shape, change their sort of structure of the tissue that's forming, changes in tissue shape, and those are all interesting things. And I'm going to change gears a little bit and talk about minimal cells and how their structure is actually useful for observing some of these collective processes. And this paper is a recently published paper in Nature Communications titled In Vitro Assembly, Positioning, and Contraction of a Division Ring in Minimal Cells. So minimal cells are a construct they use in synthetic biology, and it's usually a cell with the minimal number of genes required to turn the cell on and off, or to have the cell have basic functions. People have used minimal cells in different ways. It's interesting for looking at sort of the baseline mechanics of a cell, what it can actually produce. So in synthetic biology, they're producing a lot of compounds for industrial uses. And also looking at how it produces things like proteins. And so I don't think they've used minimal cells too much to create things like tissues or other types of things relevant to developmental biology. But why don't we read through this paper and see how it fits into what we're doing. So the abstract reads, constructing a minimal machinery for autonomous self-division of synthetic cells is a major goal of bottom-up synthetic biology. So this is like working from the genes upward instead of working, say, from the tissues down to the genes, or looking at how the tissues and the phenotype interact with the genes. One paradigm has been the E. coli divisome, with the MIN-CD protein system guiding assembly and positioning of a presumably contractile ring based on FTSZ and its membrane adapter FTSA. So this is what they're doing, is they're building, they want to assemble a contractile ring. So they want to assemble something that's going to contract along a ring, and they're using these different proteins to do this. Here we demonstrate the full in vitro reconstitution of this machinery consisting of 5 proteins with lipid vesicles, allowing to observe the following sequence of events in real time. So this is within a lipid vesicle. So usually these minimal cells are constructed within a lipid vesicle. So it has like basically a membrane, and then you can do a lot of different things. Some people have experimented with kind of the origins of life using lipid vesicles. They put a bunch of molecules in there. They add an electrical charge. They're able to reconstruct the Miller array experiment, or other types of things where you want to look at like self-assembly. So these are very useful tools. So putting these 5 proteins within a lipid vesicle allows you to observe the following sequence of events in real time. The first is assembly of an isotropic, which is having different properties along its network, filamentous FTSZ network. So these are different filaments that have different properties in the network. Its condensation into a ring-like structure, number 2, along with pole-to-pole mode selection of min-oscillators, resulting in equatorial positioning. And 3, onset of ring constriction, deforming the vesicles from spherical shape. So 3 is where you get this, or 2 actually is where you get this condensation into a ring-like structure. And then 3 is the onset of ring constriction, which is where deforming vesicles from spherical shape. So if we take a very broad view here, it could be something like this, where you have, in 1, you have these kind of this isotropic set of things here. Then in 2, it starts to organize into a ring. This. And then 3, that ring constricts like this. So it's like organizing it into like this ring-like shape. And then there's a constriction of the ring itself within that. This wasn't a very good drawing, but basically this is constricting this ring. So there's a lot of morphogenetic movement in this little group of filaments. And it's really interesting how this is all controlled by these proteins and their levels. Besides demonstrating these essential features, we highlight the importance of decisive experimental factors, such as macromolecular crowding. So these are things that you might encounter in an experiment that influence the results. Our results provide an exceptional showcase of the emergence of cell division in a minimal system and may represent a step towards developing a synthetic cell. So this is a division ring. This is where you basically are trying to put together a multiple protein system. So this is a technical challenge within synthetic biology. They talk about the self-organization of min gradient patterns and the FTSZ polymerization depolymerization dynamics that they've been able to put. They've been able to reconstitute this in vitro. They've been able to replicate what's going on in the biology in vitro, so they can control it. This is done on supported lipid bilayers and inside lipid compartments. So these are these lipid vesicles that we were talking about. So there are a number of ways you can implement this. They're doing it in a lipid vesicle, but you can do this in other places. FTSZ is shown to polymerize into dynamic ring-like structures that can deform freestanding membranes, and co-reconstruction of the min positioning system in membrane-anchored FTSZ on supported membranes is confirmed the spatial regulation of FTSZ polymers by min patterns. So a lot of this is spatially registered, and it's being constructed in this ring, and then it constricts. And then you get this division event that happens. So they talk about the multi-protein system. So this is your system here. You have the lipid membrane. You have these proteins sitting on the lipid membrane. So this membrane is at the edge of the compartment, or it's at the bottom of the compartment. And you have this sort of these chemical reactions that occur. These are the division proteins that are important. These are purified proteins, cell-free expression. So this is the lipid vesicle where you have these two things on the surface either side, and then here's the ring in the middle that forms. So you get this ring in the middle between these two side wall things where the proteins reside. So you have two poles on the lipid vesicle. You have this ring in the middle, and this is driving cell division. So this is these are almost like centromeres in an actual cell. That's what they're trying to replicate. So they have this anisotropic collection of these fragments, and then they come together into a ring here, and then the ring constricts. And then ostensibly this lipid vesicle, where the cell would divide into two parts according to these poles. So you can see that this ring here then exists in the lipid vesicle, the middle. It's acting sort of like a centromere. And then this constricts. These poles pull apart. And then you have two cells. And you have the protein complex on either side. It's a little hard to see, but I drew it. So then you can see like crowding. You have a dot pattern, a mesh pattern. There are different ways to design this. I'm not an expert at synthetic biology, but I'm very much a fan of the approach. So it's something that is a very nice frontier if people are interested in getting into that. And so they look at different crowding conditions on min wave dynamics inside lipid vesicles, which have been partly addressed in the different experimental approaches so far. So they try different things to look at that, crowding effect. So some of the results are optimizing FTSZ and min-CDE reconstitution in lipid vesicles under macromolecular crowding conditions. Another finding is co-reconstruction of min-CDE and FTSZ under crowding conditions yields the assembly of pronounced FTSZ ring structures. So they have this crowding condition. Min-C is not required for FTSZ ring positioning, so you find that there are some proteins that aren't directly involved or required for certain things, but they're nevertheless in that suite of minimal cell. They're included in the minimal cell, but we don't know exactly what things are necessary and sufficient for different processes. And so this is an example of a ring in the lipid membrane, and here's some more images of this where you see the expression of these. This is min-CD and FTSZ being co-expressed. This is min-DE and FTSZ being co-expressed. So you get a cell what it looks like. So you can see that these proteins are being expressed. They're going to form this ring. This ring then sits at the middle and contracts. This is beautiful work. Some of the staining, it just shows it so well. This is the transition of the FTS structure in the min wave. So you actually get a wave here. You get no patterns. Then you get an FTS mesh. Then you get this pole-to-pole oscillation of this ring plus the ring structure. You get the traveling wave and antagonistic localization. And you can see this again in this picture. So you get this ring that forms, and it's forming towards the middle. So it's just basically all this free protein that's coming together to organize like this. This is a pole-to-pole oscillation. So in our group, we often talk about like differentiation waves and oscillations in cells. And so this is a good example of this kind of oscillation. This is not going to result in a differentiation wave, but this is kind of interesting how these oscillations can be replicated using chemical self-organization principles. So this is a pole-to-pole oscillation of the ring. This is the static pattern of the ring. So they can do interesting things with the ring aside from like just moving to the middle. You can see things that are, kind of, it's stable, but it's not, static. Sometimes it's static, sometimes it's not. Sometimes it moves from pole to pole. And so this isn't really like cell division. Like I said, this is an analogous cell division. This is where you get, you're basically investigating the sort of the self-organizing principles of cell division and some of the things that are happening in the process of cell division. But you're doing this in an experimental system that allows you to explore some of these chemical self-organization principles, chemical morphogenesis. And so it's very interesting how they do this experimentally. And so the vesicle diameter is, if you vary the vesicle diameter and make it bigger, you get ring structures, ring deformation, no structures. You get more rings plus with deformation as the vesicles get bigger. You get more ring structures, period, as the vesicle gets bigger. So these FTSZ structures increase in terms of their frequency as you get a bigger vesicle. So it's like having a bigger space allows for more things to happen. And then you have this aspect ratio of the ring and then the diameter before deformation. So the bigger the diameter, the smaller the aspect ratio of the ring structure. You have just plain ring structures at the top with a high aspect ratio. Ring with deformation happens at probably 0.95 aspect ratio and below. And so that tells you something about, again, how this ring forms and how it remains stable. So, and then if you're interested, there are references at the bottom. So I think that's a very interesting approach. There's some, if you're interested, actually, the reference section has some good references on synthetic biology and some of the state of the art in that area. So I hope you learned something here, and thank you.
Yeah, I had him to the University of Manitoba. He used to be at Brandon University, and I had him out to do a seminar at the University of Manitoba.
Oh, okay.
Okay.
Yeah. So yeah, that's all I had. Dick said that he had talked about, or he had a dream last night about addressable X-ray sources. And I don't know if you wanted to explain that a little bit.
The idea is, this is our other project on breast cancer. We're considering using polycapillary X-ray sources which would be moved by a robot, but these things weigh 2 kilograms, and so the accurate movement is going to be fairly slow. In an addressable X-ray source, you can address what you do is you have multiple X-ray sources and you address which one is going to be turned on, and apparently they can be put in arrays. And there are about 20 papers on them, which we'll have to review. I don't know how far that technology has gone, but people are working on it. So the idea is that you just address where you want the X-ray source to come from, and presumably you can address the direction. Okay, and what this does is it gives you X-ray sources that are analogous to what are called light fields. Okay, and now there's a whole technology now of light field cameras.
Oh, light field cameras.
Yes, light field cameras.
Okay.
I actually have one.
Okay.
Yeah.
What's the
light field camera?
What's special about them?
An ordinary camera uses a lens, and it focuses the light coming from a given direction. A light field camera picks up light from all different directions, and then with an algorithm you can select what you want in focus.
Oh, okay. I think I've heard of this.
And, I have one of the first commercial ones, which is only 1 megabyte. Yeah.
Oh. What is it? What's the name of the company that made it or makes it?
I forgot. I bought it 10 years ago.
Oh.
It's sitting in the trailer, so I'm not going to get it now in this weather.
No. Okay. Is it snowing where you are?
No, it's raining.
Oh, okay. Coming right up, snow. Yeah.
At any rate, so it looks like we can have, since these X-ray sources are electronically addressable, we don't have to have any moving parts.
Right.
Yeah. Well, that helps.
I don't know how intense they are. I don't know if they can be used for killing tumors or not. So, and I don't know how accurate they are.
Well, detecting them is the first step, major first step.
That's done nowadays with single photon X-ray counters, okay? Which you can buy in arrays, so you can get a detector array that counts photons. So that's, so to speak, off the shelf. But I don't know about these addressable X-ray sources. But it's an interesting development.
Yeah. Is the light field camera, is that something that when you capture the data and you select it after it's collected digitally? Right. Or okay, so you could select a number of different views.
Right.
Okay.
Or you can select a number of different depths, things like that. Okay, they're typically used for depths. But light field is the concept of a light field. If you look around any room, wherever you put your head, you can pick up light. Right? Yeah. Okay. So a light field is all of the light photons moving around the room at once. All the different angles.
Okay.
So that's where the name comes from. So it looks like we might be able to create a light field at X-rays now, and actually have a source that produces a light field in X-rays.
Yeah.
Okay.
Yeah. Yeah, that sounds great.
Yeah, long conversation with Tom and Noor this morning.
Oh, okay. That's good.
Yeah. So I'll put the literature together tonight and upload it tonight.
Okay. Yeah, that sounds great. Yeah. Just a few words on light field imaging. So if you're interested in light field imaging, we have a few more references for you. The first one is the Wikipedia page on light field camera. So light field camera, or a planoptic camera, is a camera that captures information about the light field emanating from a scene. So it's basically picking up the light coming from a scene at various wavelengths. So it's collecting all that information. Then inside the camera, it's taking different parts of that information and processing it. So for example, you can, we'll see in a bit, you can change the focus, you can do other things, you can take the information you want out of that image. So one type of light field camera uses an array of micro lenses placed in front of an otherwise conventional image sensor to sense intensity, color, and directional information. Multi-camera arrays are another type. And then even holograms, which we're familiar with, are a type of film-based light field image. So hologram is where you have, you record a wavefront and then you reconstruct it in pieces. So you can generate three-dimensional images. So this is an example here of light field photography. This is a dog with a ball on the floor, and you've taken this light field image and you can change the focus of the image because you have all that information collected. You just need to process it. So there are different types of planoptic camera. There's the standard planoptic camera. This is a mathematical model used by researchers to compare designs. This has micro lenses placed one focal length away from the image plane of a sensor. So this was experimental in the early aughts. Then maybe about 10 years ago, it came on the market for the first time, and it's been with us ever since. I remembered an article from the IEEE Spectrum that I remember reading, but it turns out it was like a decade ago now, so I wanted to find a newer reference, and I'll talk about that in a minute. You also have your focused planoptic cameras, so you can position your lenses in different places, and you can play around with it. You can get these kinds of image effects, and you can then collect data that have like things at different focal planes. So you can see how this is useful for biological imaging as well. You can capture different aspects of the thing that you're imaging at different focal lengths and different properties of the image. We actually have that now with fluorescence imaging, but this is, of course, a little bit different animal because you can actually put these things together as part of the main image instead of stitching it together post hoc. So there are a number of prototypes that have been created. There are all sorts of people like creating prototypes of innovative ways to do this type of photography. This is very useful. This is also a area of computer graphics research. So if you're interested, you can find out more. And so there's even a desktop software, Lytro Desktop, which allows you to render light field photographs taken by Lytro cameras. So you can take the image and then render different types of scenes. So this is the article from the IEEE Spectrum from 2020, and this one actually goes into an immersive display, creating panoramic virtual screens. So this is not a primer on light field imaging per se, but this is a immersive display that uses light field imaging as its source. So just imagine wearing a 200-gram object on your face for 6.5 hours. It's really exhausting, but 6.5 hours is the average time we spend in front of computers easily every day. And so this led this inventor to come up with some level of immersion in a display that you don't have to wear on your face. So this is like merging virtual reality and this sort of light field imaging, because the information that's captured through light field imaging can be processed in different ways to make it immersive, because you have many, many different fields of view and you can render all of those. So this goes through this technology, this application here. And so whereas conventional displays direct flat images at its viewers, its light field display creates a window-like 3D scene by recreating the field of light rays that might travel from every point and in every direction within 3D space. So in a light field camera, you're capturing things from a three-dimensional space, and every ray of light that's coming, there are angles, so you have angle-sensitive pixels, you have things that are straight ahead, and then you can render it in the same way. So-called autostereoscopic displays use pixels that send light in specific directions to project a different image at each viewer, different image at each of a viewer's eyes. So you basically have you have two eyes. You're projecting things into those two eyes. And if you've ever worn a VR headset, you know that there are two cameras in there. There are two lenses that you look through. And because you have stereoscopic vision, you merge those images. In this case, you can send rays of light or pixel-specific rays into each eye, and you can actually, that can enable stereoscopic vision as well. So they have autostereoscopic and multifocal displays for 3D applications. In this application, though, they want to create a virtual panoramic 2D image and deliver it to the viewer. They call it a near-head display. So it's not a heads-up display, which is what we usually use in VR, but a near-head display. And so you can curve the display to take advantage of what you've captured in your camera and project it to the person looking at it.
Okay.
Yeah.
Thank you.
Yeah. All right. Have a good week. I guess we'll wrap it up for today.
Bye.
All right. Bye.
Okay.
Thanks.
All right. Bye.
Bye.
Bye.